Thank you so much. I'm very grateful to be among you as part of this event, and looking forward to seeing you all in person shortly. So today I'm going to give a very brief overview of the biosignatures field. Now, of course, it's a very big field, growing very rapidly. We've discussed various telescopes, missions, and technologies that are coming online now and in the future. So its rate of growth will probably increase. I'm going to survey as many of the methods in the biosignatures field as I can today, and I'm going to inevitably emphasize those which I'm more familiar with. So I'm going to sort of play to my strengths a little bit. And as such, I've roughly divided things into chemical biosignatures, which has been the main approach in the field up until recently. And then for like the second sort of 1/3 of the talk, I'm going to talk about a non-chemical approach that I've been developing with various colleagues, including Mike and Anna Ruud, and others at Caltech and beyond. So on the side of chemical biosignatures, it was sort of inspired by the NASA definition of life, which is that life is a self-sustaining chemical system capable of Darwinian evolution. So note the emphasis on chemical system, and we tend to think about life in this with this chemical metaphor because there are such a huge diversity of organic and inorganic, well, let's say organic molecules plus their inorganic extra components and ligands, which comprise life, and the complexity of the chemical reaction networks is huge and diverse. And so that has basically influenced our search for life, because it seems more or less impossible to imagine a version of life which is not sort of dominated by all these constraints from chemistry and all these molecular properties. So there are many different ways that you can use the sort of unique chemical features of life to pick it out among the abiotic background. Now, of course, a lot of this relies on spectroscopy and spectrometry, which has been raised this morning, and there are many different flavors of those techniques, like gas, for example, pyrolysis, gas chromatography, mass spectrometry is a popular one, which several of us work with, and we can use those to identify molecules and particular features. As we conduct all these searches, the way I try to think about this is that every biosignature technique, from a purely philosophical point of view, will always potentially be vulnerable to false positives and false negatives. And I think in general, we should, as astrobiologists, we should be very wary of ever saying, I have a technique which is not vulnerable to either false positives or false negatives. So these are things that we just have to live with, and we have to understand and quantify, and 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 use a plethora of techniques to try and mitigate the potential sort of false positives or false negatives of one technique could potentially be compensated for by deploying other techniques. So the as Mike discussed, when we finally sort of claim to have discovered life, it'll probably be from a multitude of techniques rather than just a single one. So to start off with, and I apologize, I'm not doing these subfields of biosignatures justice, but I'm just going to talk briefly about the most direct way of detecting life, which is where you've literally sort of found a obvious sign of life that is more or less unambiguous. So one example could be finding a DNA molecule on Mars. Now, probably a lot of us, for one reason or another, may not expect that to happen, but it's not beyond the realms of possibility. Mars was similar to Earth at the early stages of its life. So, it could be that one day either a sample which has been returned from Mars, or an astronaut or a rover on Mars digs down and just hits that lucky spot and runs some form of spectrometric analysis, for example, and finds a DNA molecule. At that point, we would probably think we've hit the money. Um, so so we can just look for the molecules that we're familiar with life on Earth and compare the mass spec data to standards and try and do things that way. Um, other possibilities which are a bit less direct are things like chiral excesses. Now, as we know, life prefers particular chiralities of molecules. So, for example, all amino acids that life uses on Earth are left-handed amino acids. And in general, the world of chemistry, not the world of biochemistry, but the sort of blind world of chemistry, pays no regard to chirality. So you tend to get both handedness molecules in 50-50 mixtures. Now, there are various ways that people have suggested by which this symmetry can be broken. So you can have, for example, autocatalytic reactions, which not only amplify themselves, but also suppress the amplification of the opposite handedness, and that can lead to chiral excess. And again, this brings us back to sort of false positives and false negatives, because there's a possibility that abiotic reactions could produce chiral excesses. It's an exciting and ongoing field. So time will tell. And related to this, there's also, of course, isotopic anomalies. Now, for things like carbon and nitrogen and several other elements, life prefers using lighter isotopes than the heavier isotopes. So we can look for isotopic anomalies. Once again, false positives, false negatives. So there was some interesting recent results on the isotopic side on Mars, but there are also potentially ways that these could be explained with abiotic means. And before I leave this slide, just to reiterate this problem of being fooled. There was this really nice paper written by our friend and colleague Laurie Barge not so long ago, where she sort of discusses all of these different sort of false positives ways that we could be tricked. So even, for example, simple cell membranes or protocells that could be abiotically formed could mimic life and look very much like life. But until you actually verify that it's a real functioning cell with a let's say a replication cycle and it's evolvable and so forth, only then may you potentially be able to say this is life. So that would be the sort of best case direct observation. In a lot of cases, we perhaps can't go to the planet. Many of us work on remotely detected data from very, very distant objects where we, as Professor Young likes to say, we only have one pixel. So we're looking at a planet with no spatial resolution, and I'll talk a little bit more about that later. But often we can get transmission spectra, so where we're seeing the light from the host star pass through the planet's atmosphere, and then there are various sort of quantum level interactions between the photons and the molecules in the atmosphere, and the photons which eventually reach our instruments can tell us something about the contents of the atmosphere. So this is another recent paper by several of our friends and colleagues where they sort of simulated what it would be like if JWST was looking at Earth from a great distance, and they conclude that in fact, you can find various disequilibrium pairs like methane and oxygen. You can even see gases like CFCs, and so depending on your perspective and persuasion, this type of spectrum would potentially be enough to convince you that Earth is a living planet, not only a biological planet, but a technological planet. So we can look at these transmission spectra and see what molecules are there and perhaps infer some of the chemical reaction networks and types of chemistry that's going on. A very interesting, complementary and new approach to this question, which is still sort of focused on the molecules and the chemistry, but actually takes tools from information theory to analyze the transmission spectra that we might get from exoplanets. So this work was done by Sarah Vanner, Marcelo Gleiser, and Lisa Kaltenegger. And what they basically do is they take exoplanet transmission spectra and compare pairs of exoplanet transmission spectra, primarily between the sort of the one we're most familiar with, which is of course Earth in the modern era, and they compare that with, for example, Earth in the distant past, or Earth if it had a different type of host star. And as we can see, we get differences in the exoplanet transmission spectrum that you would see. And you might ask, well, how can we compare this? So you could just look for the presence or absence of particular molecules. But what Sarah did here is she used some information theoretic metrics to treat these exoplanet transmission spectra as probability distributions. So there you're sort of saying, well, maybe I'm not going to focus on particular features in this spectrum. I'm going to just look at the statistical differences between one transmission spectrum and another. So we treat them like probability distributions, and the metrics that Sarah used are basically entropy measures. So she used a symmetric version of the Kullback-Leibler divergence, which specifically is the Jensen-Shannon divergence here. So it's just like a weighted version of the Kullback-Leibler divergence. And this is a way of basically, it tells you how much the knowledge of one probability distribution reduces your surprise about viewing another probability distribution. So this gives you a very nice agnostic way of comparing exoplanet transmission spectra. So in this case, we can say how different is a measured transmission spectrum from the Earth that we know and love. And this offers us sort of options in terms of comparing the complexity of exoplanet transmission spectra. Perhaps there's some correlation between the presence of life and the complexity of these spectra. Now, of course, another very prominent and promising approach, which Mike touched on a little bit, is measuring planetary thermodynamic disequilibria. So this, of course, was suggested famously by Jim Lovelock some decades ago. So the idea here is that life is a far from equilibrium system. Not only that, but it also seems in inverted commas to push its environment further from equilibrium. So not only does it make use of thermodynamic gradients, but it also creates additional ones. It's almost like thermodynamic niche construction. So while it feeds on some gradients, it creates others. And this could potentially be used to detect life under the assumption that that the more thermodynamic disequilibria we detect in a planetary atmosphere, the more likely that life is present, producing or maintaining that disequilibrium. So this has been pioneered significantly by Josh Krasanson-Totten at the University of Washington, and I borrowed a couple of his figures here, and just sort of showing how they compute some of these disequilibria between a sort of initial or observed state and the equivalent state if you let that system run to equilibrium. And you can essentially calculate a sort of Gibbs free energy difference between the two. And that gives you an estimate of the degree of disequilibrium of that planetary atmosphere. And when you apply that analysis to the solar system, you of course find that Earth is significantly higher than the other planets. Our sister planet Mars comes in second, Titan pretty high. So these are sort of encouraging results. It's sort of in line with our astrobiological sort of target list. And so on a similar theme of looking for thermodynamic disequilibrium, thermodynamic disequilibrium can be loosely associated with entropy differences. Entropy is a measure of syntactic information, and information is a key property of life. So if we look at the non-living world, information doesn't seem to play too much of a role, whereas with life, it's fundamental. So there's information stored in our DNA, there's information stored in epigenetic markers, there's information stored in the states of our neurons, in the internet. It's everywhere, and it has this strong causal influence on the world in a way that doesn't appear to be present in non-life. And so information-based tools for detecting life are becoming very popular, and there's this closely related property of complexity. Now, complexity, you could say, is a little bit harder to define than information. We can define syntactic information fairly readily using Shannon's formula. There are various measures out there that have been suggested for complexity, but it sort of depends on the context, and it's very much an emerging field. But an approach to biosignatures still within the realm of molecules and chemistry that's gained prominence recently is basically abandoning the pursuit of specific molecules as a sign of life and just looking instead for molecular complexity. So, so we can play a little game here. Actually, this may be too easy, but um, which do you think is the biological molecule out of these two? And Mike's not allowed to answer. And which is the non-biological? One is non-biological, one is biological.
The right
Oh,
is biological.
okay, very good. So you know what the one on the right is. I mean, okay. So this example has been made somewhat famous because it's Taxol, which we'll see again in a minute, whereas this one on the left is a mineral. Um, so, so the question uh, is there some fundamental association between life and complexity, and specifically the molecules that life produces and that it uses? Are they fundamentally more complex than anything that the non-living world can produce? So yeah, here we have Taxol, that's an anti-cancer drug, and here on the left we have a naturally occurring mineral. So you've probably all heard of this approach to measuring molecular complexity called assembly theory. So this came from um, Professor Lee Cronin at the University of Glasgow and Professor Sarah Walker at Arizona State University, along with many other collaborators, Stuart Marshall, Cole Mathis, etc. And assembly theory tries to quantify the complexity of molecules or objects in general by essentially counting the minimum number of elementary steps that it takes to produce that object. So you have to essentially define a priori the building blocks of your process, and then you also have to define the allowed construction steps that you can use to produce your final target object. And then in principle, you can enumerate all the possible different ways to make your object, and you find the shortest one out of this sort of large combinatorial space of possibilities for building your object. And then you take the length of that path, the path that it takes to build your object. The length of that path is taken as a proxy for the complexity of your object. Um, and the other key thing alongside the number of steps taken to make this object is what they describe as the copy number. So their suggestion, their proposal is that if we find molecules out there with a high assembly index is this measure of the number of steps taken to produce the object, high assembly index and high copy number, as in there are many of the molecules present. When you do your mass spec analysis, you can see it, as in it's not just one of the object or a small number. Then that suggests that an information processing system produced this object. And so the justification for this is so this again is taken from one of their earlier papers. The justification for this is basically the correlation between biological samples and the high molecular assembly or the assembly index. Specifically, the biological samples are able to get assembly indices of above 15, whereas all the non-biological ones tend to stay underneath. And this has led the authors to suggest that um, this this method may be vulnerable to false negatives, but should not be vulnerable to false positives because nothing non-biological can get above this 15 threshold. Now, the method has been quite controversial to say the least, and and has attracted a lot of attention from the media and from other scientists. And in particular, people from the realm of computer science and information theory have suggested that the molecular assembly or the assembly index is similar, if not identical, to some measures of algorithmic complexity from the computer science field, specifically things like the Huffman encoding, which is a lossless compression algorithm that came out in the 50s, I believe, which also tries to find the most optimal way to compress a given object, like it could be a string of data. So it's looking for repeating segments in that string of data, finding the most common one, assigning the shortest codes to the most commonly occurring blocks, and basically compressing the data by this sort of iterative hierarchical process. So yeah, there have been claims that actually this is from a previous paper. This paper has now been published. It's no longer an archive preprint. But this paper sort of claims that you can achieve the same results as assembly theory with these methods from computer science, basically from compression algorithms. And I also wanted to briefly touch on some very interesting work that's been done by Mike and Bob Hazen and Jim Cleaves and others recently on looking at the assembly index of some very, very interesting and exotic minerals. So I've just shown like a smattering of some of the really cool examples here, especially like this one called paddle wheelite, has a molecular assembly of 17. These ones Ilmajikite can have 1821. The key point here being that these are above that sort of threshold of 15. So the question is, does this actually serve as a false positive? The counter argument might be that, well, these minerals are perhaps very, very rare, so their copy number is low, or perhaps, you know, they don't occur in the absence of life. This is, of course, an interesting and ongoing discussion. Um, but but there's no doubt that this approach of molecular complexity is going to be a sort of keystone of biosignatures, or perhaps agnostic biosignatures, as time goes on. Now, I have to go back to Mike's work again. Mike, Jim, Nicholas Gutenberg, Anirudh, and others have done this super interesting machine learning study of gas chromatography mass spectrometry data. So the raw data looks rather like this. And what they're able to do is apply machine learning techniques, project everything back into some principal components, in particular the first two principal components. When you plot all the samples out as a function of those two, you find that the biological samples cluster away from the non-biological ones, and even the sort of dead biological are distinct from the alive biological. And again, we're going to see fantastic growth and development of those methods in coming years. Okay, so that's a very brief summary of these approaches inspired by chemistry and molecules. And for the remaining time, I'd like to just like that. But
Hey, Stuart, let's try to wrap up soon so we have time for discussion.
Sure. Okay, I'm going to zoom all the way through. Blah, 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 blah. Here, what we basically do is we measure the complexity of signals. In this work, we measured the complexity of exoplanet reflectance signals, and we basically found that when we modify proxy exoplanet images of Earth to reduce their surface features, so we take away clouds or we take away land, ocean features, etc., the complexity goes down. So this is something we can measure. And so this basically offers a new approach to biosignatures where we're using complexity metrics to analyze sequences, and this does not depend on particular chemical assumptions. Yeah, and you know, Jupiter's less complex than Earth, etc., etc. I will wrap up so we don't eat into our discussion time. Thank you very much for your attention.